Hello, everybody. Welcome to Pharmacology of Recreational Drugs. We got 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 25. So we got 24 people out of 50. It's almost half. Let's wait a couple minutes, see if anybody else shows up. Okay, today we're going to be talking about nerve structure and function. Nerve structure and function. Willie, you changed your hair. Like it, good.
Thanks, Lish.
We are recording. Oh my gosh. Winter Sparachin is here. Sparachin. Julia Waters. Natasha, Sean Reeser. Hello, Sean Reeser. Okay, how many people are here? 36. Why don't we go ahead and get started? Today we will be doing the next chapter, which is chapter 2. And chapter 2 is pharmacodynamics 1. So we start out talking about how neurons work, what the structure is of them and how they work. And later we'll see how we work that into larger pathways in the next chapter, or no, 2 chapters from now. Next chapter we'll talk about synaptic transmission between neurons. Okay, so nerve cells and the brain. Brain is made up of about, I think it's like 50 or 60% nerve cells, and the rest of the cells are glial cells, which are helper cells. There are 2 types of cells in the brain, your neurons and your glial cells. Glials help support the nerve cells, and they also help to in synaptic transmission. And they're finding out more and more that the glial cells actually do some information transfer as well. But I don't know that much about that topic, and it's not that important for us here. So we'll just call them support cells here. Okay, neuron structure. I'm going to go to my PowerPoint. Oops, wrong PowerPoint. Screen share. We are Santa cells, building Santa shows. We are Santa cells. Okay, so here's your neuron structure. Here's the soma, the cell body. The dendrites are at this end, and the axons over here. So what is important about this for you is the function of each of these segments here. So we'll start at this end. The dendrites receive information. Other neurons will synapse with this neuron on the dendrites. And where those synapses happen, you're going to have these dendritic spines that grow up. So that gives you a nice tight contact between the axons that are coming in and the dendrites there. So each of these little connections is a synapse. So you've got thousands of them coming into each neuron, thousands of different axons coming into each neuron, and they will synapse here on the dendrites. They'll synapse on the cell body. They may even synapse way up here on the edge where the axon is started. So dendrites are receiving information. The soma is doing 2 things. It's also receiving information. There can be synapses all across the soma, but also the soma is taking all the input together, adding it up, and then making a decision and sending that information off down the axon. So the soma then sends information down the axon, and the axon takes it away from the neuron and then synapses with other neurons and passes the information on. This is a really important thing to keep in mind here, your axon hillock. So the axon hillock is where the action potential starts. So we'll talk about action potentials today, and keep in mind there are no action potentials over here in the dendrites or the soma, but if you get depolarized enough here at the axon hillock, that'll start an action potential, and that action potential will go all the way to the end of the axon. So that's our neuron. Dendrites receiving information, soma receiving information, but more importantly, integrating it and deciding how much stimulation then the axon will get, and then the information then goes down the axon. So most neurons in your brain are going to be myelinated. Myelin is put on by these oligodendrites, which are a form of glial cell, helper cell. And what these glial cells do then is send a process out, and that process wraps around the axon, leaves a little gap here, and then another myelination and a gap. So you've got a bunch of gaps all along the way. So this is very similar to what we saw in the last class with the blood-brain barrier, where you had processes coming off the astrocytes that were wrapping around the blood vessels. This is the same kind of a thing here. The oligodendrites are wrapping around, and this is forming electrical insulation. So there's no action potential going through this myelin sheath because it's electrically isolated. But an action potential here will skip from one of these nodes of Ranvier to another until it gets over here and takes off. So basically, this is going to dramatically increase the velocity of transmittance, how fast the action potential moves down the axon. Okay, it's going to affect that. And in your brain, you probably heard you've got white matter and gray matter in your brain. The gray matter in terms of your cerebral cortex anyway, the gray matter is the outer 6 layers of cells. So it's gray because there's cell bodies there. And then the white matter is all axons that are myelinated going away from those cells. So the white matter is basically myelin mostly. That's why it's white, it's fat. Okay, we'll talk more about that. Okay, let me go back to my notes real quick just to make sure I'm not forgetting something. Axon, dendrite, body. Oh, and don't forget the cell body has everything that a normal cell body has. Every cell needs a nucleus, Rough-ER, Golgi, all of that stuff. And so all of that is in the cell body, the soma as well. Okay, so glial cells, astrocytes, oligodendrites. So we've got 2 different kinds of glial cells. Go to my notes real quick. I don't have any figures on this. Okay, so we've got astrocytes providing metabolic support. They're storing glucose. The brain needs a lot of glucose to run on. And they will talk about synaptic transmission and how glial cells can help with synaptic transmission, and astrocytes is one of those. Some neurons will release glycine, glutamate, or GABA, and the astrocytes will take them up and then recycle them back to the neuron. And they're wrapping around to forming the blood-brain barrier. That's your astrocytes. And the oligodendrites then are producing the myelin sheath for the myelinations. Okay, action potential. Okay, so we're going to start out talking about the action potential now. I will share with you these. Oh, wait, let's go to okay, we'll start here. So an action potential is basically a change in the membrane potential. So to talk about the action potential, we have to talk about the membrane potential first. So the membrane potential is set up by 2 different things. One is the fact that you've got an asymmetric distribution of ions. Oh, wait a second, I've got a chat question. Will we have to know any other neuroglia other than, no, you won't have to know. These are all the only ones I know, so you won't have to know anymore. I'm not the brain neuroanatomy specialist that I should be. So just what I'm talking about. Oh, that was a private message, Willie. Okay. So Willie Corley's asking if we have to know any other neuroglia, and I'm going to say no. Okay, back to our action potential. So set up by two things. One is this asymmetric distribution of ions. So you've got high sodium outside, 145 millimolar, and low potassium. On the inside, you've got the opposite, low sodium and high potassium. So the reason for this is because you've got this enzyme, sodium potassium ATPase, in the membrane that is pumping sodium out and potassium in. Keep in mind the cell's not going to change the extracellular solution at all. It can only control its own intracellular solution. The extracellular solution is interstitial fluid. Interstitial fluid is controlled by the blood. The blood is controlled by the kidneys. So all of that is controlled by your kidneys. What's going to be outside the cells. What's inside the cells then is controlled by the cell. So this sodium potassium ATPase, if I can screen share an animation here. Come on, come on, come on. Come on, come on, please share me. Oh yeah, I gotta share you. That's a little bit of Beatles for you there. I know how you are all Beatles fans. That's just my way of covering up the fact that I don't know what the heck I'm doing. Oh, here it is. Okay. Okay, here's our sodium potassium ATPase. So it will bind 3 sodium on the inside of the cell, pick up an ATP, phosphorylate itself, and that causes it to change into a different conformation. Sodium go out, potassium bind. When the phosphate comes off, the potassium comes in. So you can imagine this thing just constantly going. It's not being turned on or off. It's just always on. So the sodium potassium ATPase is then setting your ionic balance between the inside and outside of the cell. So you've got that. You got that going for you. So that's one of the two things that set the membrane potential. The other thing is you've got selective permeability in the membrane. So membranes are impermeable to sodium and potassium. They can't go through without a channel to help them get through. This is a structural drawing of what a channel actually looks like. This is a potassium channel, but they've got like a lot of structural alpha helices on either side. You've got a little beta sheet in the middle, and a hole or a pore in the middle of this ion structure. So this beta sheet then is a selectivity filter, which means that ions will bind to this as they go through. And that makes it so that a channel only will allow potassium to go through, or some will only allow sodium to go through, some will only allow calcium or chloride. They're all selective for one particular ion. And it's gated. And so basically the shape of this thing will determine whether it's open or closed. In the closed state, that gate is closed and sodium or ions don't go through. The open state, they can go through. And I think I've got an animation of that too. Let's see what we got here. Okay, so here's our channel in the closed state. When it opens like this, now you can see it's open down here, and then things can come through. Close it again, and there. So your ion channels have got at least 2 states, open and closed. Potassium channels just have the open and closed. Sodium channels we'll talk about with other states as well. Okay. Let's go back to the notes real quick. Okay, so back to our PowerPoint. So there are a number of different channels that can allow ions to go through the membrane. The one we'll mostly be talking about, at least in this chapter, is the voltage-dependent channel. So these voltage-dependent channels have got positive charges as part of their structure. And this is kind of weird to have a transmembrane protein that has positive charges in the membrane. As you know, the membrane is nonpolar, and charges tend to be, any kind of transmembrane protein tends to have the charges on the outside or the inside, and nonpolar amino acids in the middle, and that allows that protein to be in the membrane. Here, you've got 6 times 4 is 24. You've got 24 transmembrane regions that make up this channel. Of those 24, 4 of them are positively charged. So you've got this positive charge in the membrane, which means that when the potential of the membrane changes, it shifts to follow that change in potential, and that causes it to open. So you can see here, you've got negative charges inside and positive charges outside. And so the pore is closed. When this membrane potential flips, so here's negative outside, positive inside, these positive charges pull the structure that way. Now it's open and sodium can come through. So that's your voltage-dependent channel. And then you've got your ion-activated, or neurotransmitter-activated channel, or ligand-dependent channel. And in this channel, again, you've got your gate that keeps it closed, your selectivity filter, but what causes it to change conformation and open is the binding of a neurotransmitter. So these are what your nicotinic acetylcholine receptors are like. The acetylcholine binds, the channel changes conformation, and sodium comes through. And then we'll also talk about metabotropic receptors, which are receptors that involve a somewhat more complicated metabolism than just binding neurotransmitter and opening. Here you've got a receptor, neurotransmitter binds to it, it then interacts with another protein here. Shouldn't have done that, sorry. Interact with another protein, a G protein usually, and then that will open the channel. So we'll see a lot of these receptors as well in this class. So let's, as long as we're here, nope. Let's watch how these work. Oh, I don't know if you can't see this, I'm sorry. I have to remember to keep sharing. Sharing is caring. Okay. So here's your voltage-dependent channel. And if you've got the online book, you have access to these animations as well. So here's sodium. Sodium can't get in. When the membrane depolarizes, watch these charges will flip. Boom, negative outside. And then your channel opens, and now sodium go through. Notice how there's always sodium here. One comes in and knocks the other ones down. So you've always got sodium bound to that pore, which is your selectivity. When the cell then repolarizes, it'll go back to its original state. Okay. Are voltage-dependent or ligand-gated channels ionotropic? I don't think you would use the term ionotropic for the voltage-gated channels. Voltage-gated channels are only for action potentials. When you get into the soma and the dendrites, then we'll talk about ionotropic channels and metabotropic channels, and we'll talk about that in this chapter. Okay, this is definitely an ionotropic channel, which means when the neurotransmitter goes in, that causes opening of the channel, and then sodium goes through. So that's ionotropic, where the channel and the receptor are the same molecule, same protein. I mean, remove the neurotransmitter, and it goes back. Add the neurotransmitter. Remove the neurotransmitter. I can do this all day. Okay. The direction of the channels change depending on the equilibrium within the membrane. Oh, good question. So Matt, okay, I won't say your name because it was a private message, but there was a question here that says, can the direction of these channels change depending on the equilibrium with the membrane? The answer is yes, but not physiologically. Okay, so physiologically, you'll always have sodium high outside and sodium low inside. So sodium will always go into the cell. But you can take a patch of membrane from that cell, or even the whole cell, change the inside and outside concentrations of sodium, and have sodium go out. Okay, so it's possible that they are reversible, but only, not in physiological situations. Okay, and then the last one then is your metabotropic receptor. Add neurotransmitter, boom, activates receptor, receptor activates G protein activates channel. And now we've got potassium going out. This will shut itself off. Boom, boom, boom. And it will keep turning itself back on as long as you've got neurotransmitter. So that G protein shuts itself off, and then it gets reactivated. Okay. So let me check my notes real quick, but I think that's basically the 3 different types of channels that I talk about in this chapter. Okay, so we've got our Volch-Bennett channels, ligand-gated channels, and metabolically activated channels. Okay, those are 3 different types of channels we have here. Now your inotropic and your metabotropic channels, the second 2, will all be in synapses. And then your Volch-Bennett channels are only in the axons for passing the action potential. So how do these voltage-gated channels cause this action potential to happen? Well, first we'll start with the resting membrane potential. So, oops, I don't know what I'm sharing now. Let me make sure I'm sharing the right thing. Okay, this is how you get your action potential. So you've got your membrane potential. So what's going to control the membrane potential is which ions are moving through it, okay? Normally, your Volch-Bennett sodium channels are all closed. Okay, these blue ones are sodium channels. They're closed, sodium's not going through, so sodium doesn't affect the membrane potential much at rest. At rest, you've got these voltage-dependent potassium channels that are also closed. They're not having much of an effect, but what does have an effect is the leak potassium. So there's always potassium leaking out of the cell at a very low level. So this amount of potassium is a lot less than what you'll see when these suckers open, but there's always potassium leaking out, okay? So because you've got potassium leaking out, you end up with a negative charge on the inside, right? Positive charges are going out, they're leaving negative charges inside. So the inside of your membrane is negative at rest compared to the outside. And with a neuron, it's typically about minus 75 millivolts or so. So that's your membrane potential, okay? It's set up by 2 things. One is these driving forces of sodium and potassium. Sodium's high outside, it's got a driving force to come in. Potassium's high inside, it's got a driving force to go out. So you've got those driving forces sitting there waiting. Now what's going to let them go is the ion channels. Here at rest, you've got potassium channels leaking out a little bit, it's leaving, and so that's making the inside of the membrane negative. Now, to talk about the action potential, we have to talk about the different states of the sodium channel. So when the membrane is at rest, it's in this closed state, and we've got 2 different things here that are controlling movement of sodium through. One is the voltage gate, and the other is the inactivation particle. So at rest, voltage gate is closed, sodium can't go through. Now, when this patch of the membrane, say this is in the axon hillock, right? Some depolarization comes from the soma and hits that. The sodium channels are going to see that depolarization or change in voltage, and that voltage gate will open. Okay, just like we saw before with the channel doing this when the voltage changed. It opens up like this. Now you've got this big hole in the membrane. Sodium comes flooding in. That's only going to last for about 2 or 3 milliseconds. And and at about that time scale, this inactivation particle is going to swing up and plug that hole. So at this point, no sodium goes through. Now, both the inactivated and closed, there's no sodium going through, open there is. But there's a big difference between inactivated and closed. Here in inactivated, it can't open again. There's no way to get that inactivation particle out unless the cell repolarizes first and then waits a little bit of time. So here in this inactivated state, this membrane will repolarize because of our potassium channels. I'll talk about that in a second. And when the voltage changes back to the way it was, and takes about 3 to 10 milliseconds, that particle will come back out. And because the voltage is normal again, the voltage gate closes. Now you're back in the closed state. So the sodium channel has to go around this cycle like this. At rest, it's closed. Voltage comes, it opens, and then it automatically inactivates a short period of time later. When the membrane repolarizes, it goes back into the closed state, and now it's available to be opened again. In this inactive state, it's not available. Let's look at that animation. Okay. Brian Jacobson. Hi. I'm just saying hello. Just saying hello. Okay. Okay, so here's our voltage gate channel. Watch these charges here at the top and bottom. At this point, this is resting membrane potential. Potassium's leaking out, so it's negatively charged inside. When that flips, your voltage gate opens, sodium goes through, and eventually it goes inactive. When the membrane flips back, it goes back to its closed state. Closed, open, inactivated, back to closed. Okay, so that's your voltage-dependent sodium channel. Yeah. Okay, so what you say, Dr. Slish, Smarty Pants, you think you know so much. How does that give us an action potential? Glad you asked. Okay, so in the membrane, we've got two different channels that are going to be important for this action potential. There are other channels, but these are the two that are important. Let me pull up my PowerPoint again. Okay. Just go through it like this, and then I'll come back. Okay, so you've got two different channels that are going to be important. Your voltage-dependent potassium channels here, and your voltage-dependent sodium channels here. So at rest, these are both closed. And remember, the only thing happening is you've got that leak potassium going out. Now, a little bit of depolarization comes to this patch of the membrane. We'll talk about that, how that happens later in this chapter. But for right now, just say this membrane potential goes from -75 on the inside to like -50. So it's a little bit less polarized than it was. When that happens, your voltage-dependent sodium channels go from closed to open. Now you've got sodium rushing in, a ton of positive charge coming in. The inside of the membrane turns positive, and that's what we see here in the graph of the action potential. Sodium comes in, and the inside of the cell goes positive. Now we're all the way up to +40. So we start at -70, we're up at +40. Okay, now like we said before, with the function of the sodium channel, this inactivation particle is going to swing over and shut that channel. At the same time, the change in voltage is going to open these potassium channels. So these potassium channels are delayed a little bit behind the sodium channels, and the sodium channels are kind of causing them to open here. So when that happens, now you've got a ton of potassium going out and no sodium coming in. So all these positive charges are leaving. That brings you all the way back to the negative end of the spectrum. And here you can see you're even farther down below than you were originally because there's a lot of potassium going out. And these are, and it's more than the leak potassium, so you go even further negative. And, okay, Sean, I'll answer that question in a second. So, and then to go back to rest, that potassium channel closes. Now, again, the only ion moving is that potassium going out the leak channels, and so it comes back up to that resting potential, resting membrane potential. So just to go over that again, okay, sodium channels open, you depolarize. Sodium channels inactivate, potassium channels open, you repolarize, then the potassium channels close, and you go back to rest. Okay, you said that the amount of sodium potassium extracellularly stays about the same. Why does the membrane gain a negative charge when the sodium gate opens? Good question. Okay, so the total amount of ions outside and inside the cell don't change much at all. Okay, but what is changing is those ions right at the membrane. So all of this is happening right at the membrane and not really inside the cell or outside the cell. So there's really not that much moving, but enough to change the voltage right there. Now you could, if you had a neuron, you could shut off your sodium potassium ATPase so that you couldn't, like normally, these sodium come in, sodium potassium ATPase kicks them back out. You could shut that sodium potassium ATPase off and keep getting action potentials because you're not really moving that much sodium. Eventually, it would run down and you wouldn't have that sodium gradient anymore. But in the short term, there's not a lot of ions actually moving. Okay? Okay, so let's look at the animation of this. Sometimes these animations are a little bit too fast, I think, but they're in your book. You can see them there if you want to. So in this animation, I've put that last figure right next to the other figure, so you can see them happening at the same time. Again, I think this one may go too fast, but watch this happen anyway. It's down here, we're at rest. Sodium channels open, sodium comes in, potassium channels open, potassium goes out, potassium channels close, boom, you come back up. Okay, let's try that again. I should just loop this and let it keep going. Okay, go a little fast, but does it make sense? Okay, put your video on for a second. I want to see your faces. Your beautiful faces. Everybody coming up. Okay, so how many people by show of hands understood that? Okay, good. We're doing good. All right, we'll move on then. Okay, what's next? What is next? What is next? Let's see. Thank you, Alexis. You're humble and lovable. Next, just checking my notes just to make sure I'm not leaving anything out that's important. Okay, how does this thing start? Nothing, passive spread. Just want to make sure I don't do too much because we have to have a lecture on Wednesday too. Oh boy. I think I've covered most of what I usually cover in this second lecture. Okay. Only a half hour. Should we call it a day?
Yes, we shall.
Winter, you go.
Yeah.
Okay. All right, then. Why don't we just call it a day? Go ahead and read ahead, and then in the next class, we'll talk about how that action potential propagates down the axon and how the soma adds up inputs and activates that action potential and controls the frequency. So, all right. Nice talking to you guys. Meet you back here on Wednesday.
Before you leave, Dr. Slish,
Yep.
my boss totally screwed up my schedule for the week.
Okay.
So I won't be able to join in on Wednesday,
Okay.
but I'll definitely watch the recording. So I just wanted to let you know that.
Okay. No problem. I will definitely put the recording up.
Okay, great. Thank you so much.
Sure. No problem.
Okay. Bye.
Okay. So Serena, so all the notes are in Moodle. Okay. So if you missed anything, you can get it there. Okay, guys. Hasta luego.